Hello, welcome back. Hope you guys had a great break, and today we're going to talk about DNA replication. So the first thing we need to talk about is, when does DNA replication happen?
Well, so remember last chapter, we did cell cycle, mitosis,
Yep.
cell division. And remember we said the three parts of the cell cycle were interphase, mitosis, cytokinesis, right? So when we talked about interphase, that's where the cell spends 90% of its time. And in interphase, you have S, which is synthesis, and that's when DNA replicates.
Right, so it's G1, S, G2.
Mhm.
And synthesis and synthesizing a new strand of DNA or making a DNA
Making a
strand.
copy.
The next question I have, though, is that we know that the cell cycle happens and we have the S phase, and that's when DNA is synthesizing. But where in the cell is that happening?
Well, I'm pretty sure it's in the nucleus, because the nucleus controls all of that. Is that correct?
Right, and that's where the DNA is located.
Right.
Nucleus. So
Good.
that would
Okay.
make sense.
Good.
All right, guys, so let's actually get into the meat of DNA replication now. So when the DNA begins to split, it does it at the point that's called the replication fork. And so that's just the place where the DNA is splitting into two, because remember, it's a double helix, so it's two strands. So we got to break those apart. And then what is the bond that joins those two bases, like adenine and thymine or cytosine and guanine?
So it's a hydrogen bond.
Right.
And it's a weak bond, so
Right.
it can break.
So it's easy to break, right? And so
Right.
we have an enzyme, and we know enzymes speed up reactions, and
Right.
the enzyme here is DNA helicase.
And why we call it a helicase? Because it's a helix, right? And it's
Right.
breaking the helix up. It's an ASE, it's an enzyme, so it's an enzyme that breaks up the helix, helicase.
Woohoo! I mean,
Yeah.
gosh, science makes sense.
Yeah, I know,
So
it's a
weird.
lot.
Okay, and so the last concept we want you to understand is that DNA replication is semi-conservative. And so what that means is that the parent strand, so the DNA broke up into two parts, and each side is an old strand, and a new one will come and add to that. So it's semi-conservative because it has an old and a new. So yeah, anything else?
Nope, that's it.
All right,
That's
so
perfect.